PCR, an abbreviation for polymerase chain reaction, is a technology used to amplify specific DNA sequences and is applied in a wide range of fields. During the COVID-19 pandemic that began in 2019, PCR became widely recognized as a tool for diagnosing infections, further popularizing the term PCR. In this video, we will explain the basic principles of PCR and introduce several techniques derived from this method. Developed in the 1980s, PCR rapidly gained popularity as a method for amplifying specific DNA sequences. Before its development, DNA was amplified by introducing it into bacteria such as E. coli and allowing the bacteria to grow. However, this method was highly labor-intensive and far from ideal. Additionally, mutations could occur during bacterial growth, potentially altering the DNA sequence and compromising reliability. The greatest advantage of PCR is that the entire process can be performed in a test tube, allowing it to be completed quickly and conveniently. Today, PCR is so well established that it can be performed easily, even by those without extensive training. At the core of PCR is an enzyme called DNA polymerase, an essential enzyme for all living organisms that is responsible for DNA replication. During DNA replication, the double-stranded DNA is first separated into single strands. Then, an enzyme called primase synthesizes primers, which attach to the single-stranded DNA. DNA polymerase then reads one strand while synthesizing the complementary strand. Because the base pairs A and T, and G and C, are complementary, reading one strand automatically determines the sequence of the other. This process doubles the DNA with each cycle. The mechanism of PCR closely mimics this natural process but is carried out in vitro. The most significant difference is how the DNA strands are separated. In living organisms, this is done by enzymes, whereas PCR uses heat. At high temperatures, the double-stranded DNA separates into single strands in a process called denaturation. The temperature is then lowered to allow short DNA fragments called primers, which are complementary to specific sequences, to bind to the DNA. This step is called annealing. Finally, DNA polymerase extends the primers by synthesizing a new DNA strand, a process known as extension. Repeating this cycle exponentially amplifies the target DNA in a short period. One remarkable feature of PCR is that changing the sequence of the primers allows amplification of various DNA regions. While synthesizing long DNA sequences chemically can be costly, primers consisting of approximately 20 bases are relatively inexpensive. Once the primers are designed and all necessary reagents are combined, the PCR machine automates the denaturation, annealing, and extension steps. This simplicity revolutionized genetic engineering, earning its inventor, Kary Mullis, the Nobel Prize in Chemistry in 1993. PCR has a wide range of applications, including genetic research, pathogen detection, forensic DNA analysis, and evolutionary studies. In particular, PCR is invaluable in diagnosing infectious diseases by amplifying and detecting pathogen DNA, enabling early identification of infections. Let us delve deeper into how PCR has been used in diagnosing COVID-19, the virus that caused a global pandemic in 2019. For diagnosing COVID-19 infections, a technique known as real-time PCR is employed. Since COVID-19 is caused by an RNA virus, the process first involves reverse transcription to convert RNA into complementary DNA, cDNA, followed by PCR. However, for simplicity, this step will be omitted here. Real-time PCR enables quantification of the initial amount of the target sequence in a sample. Infection status is determined by analyzing the threshold cycle, QUART, value obtained from the amplification curve. A low QUART value indicates a high amount of viral RNA in the sample, suggesting a higher likelihood of infection. Conversely, a high QUART value suggests a lower amount of viral RNA, which could indicate mild infection or that the sample is near the detection limit. Thus, PCR has become an indispensable tool in modern medicine, playing a crucial role in supporting our daily lives.